I was just reading the latest clinical data on chronic stress and cognitive aging, and it changes how we should evaluate the biological mechanism linking emotional experience to measurable brain changes. Most discussions of stress focus on the feeling itself, the anxiety and emotional weight. But the real story happens at a deeper level where hormones are reshaping the physical structures inside your brain. The stress response is fundamentally biological. When threat is perceived, your body activates the hypothalamic pituitary adrenal axis, a cascade of gland activation that produces cortisol and glucocorticoid hormones. This is not a flaw in your design. Acute stress mobilization is adaptive. The problem emerges when this system remains chronically activated, continuously flooding your system with hormones designed for short-term survival. Cortisol is the primary glucocorticoid hormone released in response to stress. It enters the brain and binds to receptors throughout neural tissue, with particularly high receptor density in a specific region called the hippocampus. The question is not whether stress affects mood. The question is what happens when stress hormones act directly on the hippocampus, the structure responsible for converting experience into lasting memory. The hippocampus is essential for memory consolidation and spatial navigation. It is where individual experiences become woven into your autobiographical narrative, where information becomes lasting knowledge. Now, when cortisol chronically binds to glucocorticoid receptors in this structure, it initiates a cascade of changes that directly alter the physical architecture of hippocampal neurons. The mechanism can be separated into specific neurobiological changes. The first is alteration in dendritic branching, the fine extension of neurons that form the majority of synaptic connections in memory circuits. Under chronic glucocorticoid exposure, these fine dendritic branches shrink and retract. The connectivity that encodes memory and supports learning is literally being dismantled from within. The second mechanism is inhibition of neurogenesis. The hippocampus is one of the few brain regions that continues generating new neurons throughout adult life. Chronic stress directly suppresses this process. The loss of capacity to generate new neurons is a specific kind of cognitive vulnerability. It means your hippocampus loses its ability to refresh, to recover, to build new memory circuits. Chronic exposure to stress hormones, particularly glucocorticoids, has measurable impacts on brain structures involved in cognition and mental health, with the hippocampus being especially vulnerable due to its high density of glucocorticoid receptors. The supplied clinical evidence supports a narrower conclusion. Not all stress causes cognitive decline, but sustained high cortisol levels do produce measurable biological changes in brain structures that support memory. The HPA axis itself is evolutionarily ancient. It exists in vertebrates across the entire evolutionary tree, indicating its fundamental importance to organism survival under threat. The hypothalamus at the base of your brain monitors threat signals and initiates the cascade. It releases corticotropin-releasing hormone, which stimulates the pituitary gland to release adrenocorticotropic hormone. That hormone, in turn, stimulates the adrenal cortex to produce and release cortisol into the bloodstream, completing the cascade that mobilizes resources for survival. This is elegant biological engineering for acute crisis. Cortisol mobilizes stored energy, suppresses immune processes that can wait, sharpens sensory awareness, and allocates cognitive resources toward immediate survival threats. When the threat passes, cortisol levels should decline, the system should reset, and normal resource allocation should resume. The problem emerges when threat signals never fully resolve. Chronic stress means the threat signal remains active, cortisol stays elevated, and the resource allocation system remains locked in crisis mode, sacrificing long-term maintenance for short-term survival. The hippocampus is a non-essential target for this resource allocation crisis. Survival does not require forming new memories or maintaining learning capacity. Memory consolidation can wait until the emergency passes. But when the emergency never ends, the hippocampus never recovers. The crisis allocation becomes permanent. Dendritic branches remain retracted, new neuron generation remains suppressed, and memory-dependent cognitive capacity gradually declines. When you look at the proposed cellular mechanism, the key limitation is what happens at the transition from acute stress adaptation to chronic stress pathology. The hippocampus has some capacity for recovery if cortisol levels decline and stress resolves, but there may be a point of no return, a critical threshold beyond which structural damage becomes permanent. Prolonged glucocorticoid exposure can cause decreased dendritic branching, alterations in synaptic terminal structure, and inhibition of neuronal regeneration within the hippocampus, creating a biological pathway from chronic stress to cognitive decline. This is not a simple dose-response relationship. The same cortisol elevation may produce different hippocampal consequences depending on the duration of exposure and the developmental stage at which exposure occurs. The brain's response to stress is context-sensitive. A single stressor over a brief period activates different cellular responses than the same stressor repeated chronically. Chronic stress also alters the sensitivity of the HPA axis itself. The system becomes less responsive to negative feedback, meaning even reduced stressors fail to shut down the cortisol response adequately. This creates a self-perpetuating cycle. Elevated cortisol damages the hippocampus. The damaged hippocampus becomes less effective at regulating the HPA axis. The HPA axis remains dysregulated, keeping cortisol chronically elevated. Breaking this cycle requires both stress resolution and hippocampal recovery. The first without the second leaves residual HPA dysregulation. The second without the first leaves the damage-inducing signal active. The measurable outcome of this mechanism is cognitive decline. Aged humans with significant prolonged cortisol elevations showed reduced hippocampal volume and deficits in hippocampus-dependent memory tasks compared to normal cortisol controls. This is not an abstraction. Hippocampal shrinkage directly correlates with measured memory performance deficits. The structural change has behavioral consequences that can be objectively measured. Do not imply all stress is inherently damaging. Acute stress can be adaptive. Do not suggest irreversibility without qualification. These distinctions anchor the conversation in biological reality rather than catastrophic framing. This is not a miracle intervention narrative. It is a mechanistic pathway. Understanding it requires accepting that the biological system does not always recover on its own timeline, and that prevention may be more effective than reversal. The supplied clinical evidence demonstrates a measurable mechanism linking chronic stress biology to measurable cognitive outcomes operating through a specific neural structure with well-characterized glucocorticoid sensitivity. A limiting factor is the research model itself. Most of the foundational autopsy and animal model work was conducted in specific populations or laboratory conditions that may not fully predict human cognitive aging across diverse populations and life contexts. Stress responses vary by demographic, genetic background, prior trauma history, and access to recovery resources. A mechanism supported in controlled research may express differently across human populations. Do not present animal model findings as directly equivalent to human outcomes. Do not imply universal causation. These qualifications protect against the very overconfidence that produces oversimplified interventions. The important limitation is what happens when someone has pre-existing hippocampal vulnerability. Prior trauma, genetic predisposition toward HPA dysregulation, or early developmental stress can amplify the effects of new stress exposure. These individuals are at higher risk for stress-related cognitive decline even at lower absolute cortisol elevations. The mechanism operates the same way, but the baseline vulnerability changes the threshold at which pathology emerges. What does hold is the mechanism. Chronic cortisol elevation produces measurable dendritic retraction, inhibits neurogenesis, and reduces hippocampal volume in both animal models and human populations under study. What does not hold is any protocol that claims to reverse established cognitive decline simply by reducing stress without accounting for the neurogenesis recovery time and the individual factors that determine vulnerability in the first place. So how does stress actually ruin your brain? The answer requires understanding that stress is not just a feeling. It is a hormonal cascade that directly rewires the structure responsible for memory and learning. The key insight is that acute stress activation is adaptive. The same mechanism becomes pathological only under sustained activation. The distinction between adaptation and pathology lies in duration, not in the existence of the response itself. Chronic elevation of cortisol creates a cascade starting with glucocorticoid receptor binding in the hippocampus, proceeding through dendritic retraction and neurogenesis suppression, and resulting in measurable hippocampal volume loss and cognitive performance deficits. The magnitude of cognitive decline is not determined by the stressor itself, but by the duration of cortisol elevation, the individual baseline vulnerability, and the available recovery time once stress resolves. This differs fundamentally from a cause and effect narrative where a single stressor produces a predictable outcome. It is a complex systems response where context determines consequence. The practical question is whether the effect is clinically meaningful under the studied conditions. A measurable change is not automatically a clinically significant problem. The research shows that significant chronic cortisol elevation correlates with measurable cognitive decline in aging populations. This is clinically meaningful. The distinction matters for how you evaluate stress reduction interventions. A stress reduction strategy that merely reduces perceived emotional stress without actually lowering cortisol production may provide psychological benefit without addressing the hippocampal mechanism. This does not mean psychological strategies are worthless. It means the mechanism-based evaluation requires understanding what is actually being modified and whether that modification addresses the cortisol-hippocampus link. If you want to protect your cognitive capacity from stress-related decline, the priority is identifying which sources of activation are controllable and which require adaptation because they cannot be eliminated. From a cellular perspective, the goal is preventing sustained elevated cortisol. This may require addressing the source stress, improving stress tolerance, or both simultaneously. Measure what is actually supported by the supplied evidence, not how you feel, but whether hippocampal-dependent functions like memory encoding and spatial learning remain intact. Separate the mechanism from the outcome. The mechanism is well characterized. Chronic cortisol damages hippocampal structure. The outcome depends on many additional factors, including baseline vulnerability and recovery resources. Recognize that individual responses to stress vary based on genetic background, early life experiences, and current biological state. A protocol effective for one person may be insufficient for another. This episode is educational interpretation of established neuroscience and supplied research findings about chronic stress and hippocampal biology. It is not individualized medical advice. Significant stress or cognitive concerns warrant conversation with a qualified professional. The goal is understanding the biological mechanism accurately enough to evaluate stress-related health claims with appropriate skepticism and enough nuance to distinguish mechanism from outcome. Cognitive capacity is not predetermined by stress exposure. It is influenced by stress exposure, but recovery is possible, prevention is achievable, and the biological mechanisms are comprehensible enough to guide realistic decision-making. Follow the evidence boundary and separate the mechanism from the outcome. The biology is precise, and that precision deserves conversation that matches it. Optimize the protocol, protect your brain, and I'll see you in the next data set.